This is Sensus Cultural, and we are delighted to have Dr. Alessandro Prigione as our guest today. Dr. Prigione is a member of the board of directors at Sensus Cultural and a professor of general pediatrics at Heinrich-Heine-University in Germany. He is a renowned expert on various forms of intellectual disability, and we are proud to say that he's also team leader of our latest project, which we have just launched and are seeking corporate and individual funders for. This important research project, which engages world scientists from Heinrich-Heine University and the UC Davis Mind Institute in Northern California, promises groundbreaking treatments for autism and other important degenerative disorders. It is called Gene Engineering Approaches to the Treatment of Fragile X-Associated Neurodevelopment and Neurodegenerative Disorders. We are very pleased to welcome Dr. Alessandro Prigione. How is this latest research project with Sensus Cultural, which also involves CRISPR studies for altering DNA sequences and modifying gene functions, so unique?
Thank you very much. I'm very happy to be here today and tell you about our project, which is entitled Gene Engineering Approaches to the Treatment of Fragile X-Associated Neurodevelopmental and Neurodegenerative Disorders. So as you can see from the titles, there's gene engineering approaches that we propose to carry out. CRISPR-Cas is a method that can be used to cut precisely in the genome and make modification. And this can be done to correct a specific gene or to introduce if someone wants to model a specific gene mutation. So in this project, we will use such approach in two ways. First, we will use it to make a model system. And this is my part. So in my lab, we work with induced pluripotent stem cells, which are stem cells that you make from patient material like fibroblasts that we reprogram to stem cell. And then this is what we will do. So we will get patient material that we will like fibroblasts, so from a skin samples that we reprogram to stem cell. And then with this stem cell, we will use genome engineering to correct the mutations. And this will allow us to have a model system of the disease that has, it's called isogenic. That means iso is the same genome. That means it's the same patient-derived cells, but just without the mutation. So then these cells, with or without the mutation, they can be easily compared because all the rest of the genome is the same. And so then this is a model system that we can use then to make neurons or in three-dimensional structure called brain organoids that we can then use to study the disease and to understand what are the phenotypes that can we see in culture. And the second way how genome engineering approaches is used in our project is instead as a gene therapy approach, and this is the part that is being done by Paul Hagerman and UC Davis, California. And so for this part, I will leave him to describe this to you.
Will the study involve human cell samples, and is this the first? And is it based on testing on animals?
So as I mentioned, in my lab, which is located at the University of Heinrich-Heine-University in Düsseldorf, Germany. So we do not use animals. So we are part of the so-called 3Rs laboratories. The 3Rs are reduce or refine or replace. These are the 3Rs, animal work. So, and we are in the replace part. So we try to use system that let us, that allow us to not use animals. And so, and the system that we use, like I described before, is based on induced pluripotent stem cells, so iPS cells, they will directly derive from patients. So to answer your question, yes, we will use a patient material, then we will obtain through collaborators, but also from Paul from UC Davis, and this patient material, we will use it to make iPS cells. So we think that using this animal model system, but also can allow us to see things that maybe in animals cannot be seen. And then because there's a lot of difference between a mouse and a human, and especially for diseases that are associated with intellectual ability, like autism or other pediatric neurodevelopmental diseases. These are very difficult to model in an animal. So really, from the scientific perspective, using such human stem cell based models can allow us to study a process that in animal is difficult to have. So animals do not show clear signs of autism because they're difficult to be measured in an animal. So we think that by using iPS cells, we can then have a model system that it's let's say innovative, but also can help us to address questions that maybe are difficult to be addressed in an animal.
Dr. Prigione, explain to us, please, your role in helping lead this study.
So, like I mentioned before, we are located in Düsseldorf, Germany, and our expertise is really on using iPS cells, so induced pluripotent stem cells, that we generate from patient material, so from skin cells. And our focus is on using these cells as a platform to understand neurological diseases. So we take these cells, we differentiate them into neurons in 2D or into brain organoids in 3D. And brain organoids are structure that can recapitulate some of the feature of a developing brain. I said some because, of course, it's still a model. It's not a full brain. So some features of the brain are not present. So the vasculature, so the fact that blood comes into the brain, this is difficult to be reproduced as yet in the organoids, or other cells are not. So features of the blood-brain barrier between blood and the brain are also not well easily reproduced. But other aspects related to neuronal development and to how the brain generate connection and specific layering of different cell type in the brain. This can be studied, and these are supposedly important for neurodevelopmental disease. And so we think by addressing this aspect of how the brain develop in this model system, we can start to understand how Fragile X affect this brain development, and there we can identify targets for treatments, and we can test also gene therapy treatment that Paul is developing. So this is really a platform for understanding disease and looking for drug targets, testing drugs, testing gene therapy. And so that's basically how this iPS cells model, we use it, and how we'll be using this project.
Will the study be promising for a cure for Fragile X and autism?
Yes. So in my lab, we are interested in studying in using this technology and this approach of iPS cells to study diseases that are affecting mitochondrial function. So we're very much interested in finding cures for these diseases. In particular, we have current studies on Lee syndrome, which is a mitochondrial disease, and a Huntington disease. And so we're not per se expert of Fragile X syndrome, and that's why we team up with Paul. So we bring our platform with iPS cells and genome editing and differentiation in neurons and organoids, and the characterization of mitochondrial properties. So we hope that using the approach that we're already using for mitochondrial diseases and Huntington disease. We hope that we can also help the field of Fragile X. And to answer your question, yes, we think that this approach can lead to identify drugs, because that's what we're doing in the disease that we're currently studying. We perform compound screening, we identify drugs, and there is already one drug that we identified and now has been approved by European Medicine Agency for Lee syndrome. So we think that this approach can be applied also for Fragile X.
And finally, is there anything about this project that you would like to provide additional information on?
Our role is mainly to provide the technology using this iPSL technology for mitochondrial diseases. So we have an interest also in expanding on building up on this technology to other diseases. So I'm happy to team up with Paul Hagemann, who has expertise on Fragile X. And so we think that together, by bringing these two expertise on the disease and the gene therapy on his side, and on the iPS cells and brain organoids and mitochondria on our side, we think that we could hopefully bring something new to the scientific field, and hopefully develop test treatment strategies that can then be developed further in the clinic.